sensory input, right? So from the beginning, through motor output going through the middle black box, okay? So let's get started with the sensory input. So how do you guys think that flies do it once they come across each other? How do they recognize themselves? So there is a male that is walking around and comes across another fly. So how does the male figure out whether that other fly is a male or a female? Okay, Ivan, yes, that is one of the cues. Chemical signals, yeah. So chemical signals, anyone has any other contribution? No. Let's say that Antoni suggests behavior, Laura movement, which would be kind of similar, no? How they move, how they behave. Okay, so let me comment. So behavior and movement, if anything, that would come through visual. Through the visual system. The visual system has a little bit of input in recognition of a kind of specific individual, but not that much, and it's unclear how it contributes. Okay, it's not that a male walks different than a female. Okay, it's not that clear that there will be something that will help the animal distinguish. Okay, chemical signals, right? So I ask more questions because these chemical signals could be divided into different types, or detected through different senses. Okay, so chemical signals could be olfactory, like pheromones. Or they could be, how do you call it, pheromones and, volatile compounds, right? They can be detected through taste and through touch. These chemical signals that Ivan was mentioning can be detected through two sensory systems, touch and smell. Okay, Laia says wing sounds. So there is an acoustic component, okay, but that is not going to happen necessarily at the first encounter, because like flies walk around and they don't necessarily move the wings all the time, okay? But certainly during courtship, the song allows the female also to recognize and verify that the fly that has in front is a male that's trying to court her. Okay, phenotype, I'm not very sure I understand the phenotype, how they look like. Flies that are from the same stock that have the same markers, they look the same. And I'm not sure if the visual stimuli can really distinguish, whether the animal in front is a male or a female. Because if you look, they are not that different, males and females. I don't know if the male is going to go and look if the female has sex combs or not, right? That's kind of small. And by size, well, yes, females normally are bigger, but, that would mean that male needs to know that he's small, right? And that the other one is big. Anyway, the answer here is basically chemical signals that are detected through the olfactory and the gustatory systems of the fly. Okay, so the olfactory system of the fly, so there are odorant receptors that send different types of substances that are housed in the antenna. Okay, and then there's also gustatory receptors in the mouth of the fly, but also in the legs. Okay, remember that the male taps the female. Okay, so different pheromones are detected through different systems. Okay. Okay, so we have this sensory input, and now let's go and pay attention to a particular sensory input that is going to set the tone a little bit about the things that we're going to be discussing from now on. Okay. So here we have the olfactory system, and this particular compound is called CVA, cis-vinyl acetate. It's a male-specific pheromone, okay, that activates the odorant receptor 67D, that is this one here. All these green little dots are the cell bodies, right, of olfactory neurons that express this receptor, and this receptor binds CVA, okay? So CVA binds to the receptor that depolarizes this neuron, and this neuron sends a signal through the circuit, okay? So there is a male walking around, comes across another individual, an individual that we're going to call fly B, okay? So there's our fly that walks around, comes across fly B. If this fly B is a male, it is going to smell like CVA, right? So our fly is going to smell CVA, and it's going to say, oh, the individual B I have in front, it's a male. Okay, so I'm going to inhibit courtship, and maybe depending on the situation, I'm going to be aggressive to this other male. Okay. What happens? There's a female that is walking around and comes across an individual B, and this female CVA from this individual B binds to the female or 760C receptors. Okay. And what happens is that this female says, oh, this individual B that is in front of me, it's a male. I'm going to let him court me. Okay? Elena, do you have any question? Okay, no problem. Since there was like something, I was not sure. Okay, is this clear? Is this clear here? Okay, so we're going to have that a particular pheromone, that it's a male pheromone, binds to the same odorant receptors in males and females, because both males and females have this odorant receptor. Okay, so what I said is that we have I don't think I talked about a fly that was a bee fly that was female. Okay, so I said here we have a male fly that is walking around. Okay, comes across a fly bee. This fly bee, could secrete CVA. Okay, so if it secretes CVA, our fly that was walking around is going to think, oh, this fly bee that I have in front of me, it's a male. I'm not interested in courting. And depending on how the situation is, I'm going to be aggressive. What happens here is that we have a female that is walking around and comes in contact with a fly bee. Okay? So if this fly bee secretes CVA, right? It's going to be a male, right? Secretes CVA because CVA is male specific. Okay. Our female is going to say, oh, CVA has bound to her OR67D receptors, and these neurons send a signal to the brain, and her behavior is going to be, okay, I'm going to stay around and let this male court me. Okay. Maria say yes. Okay, so Laura, did you get it? Okay. So Maria, yes, that's the question that is going to come afterwards. Okay, so how is it, right? How is it that the same stimulus, right, is eliciting different behaviors in males and females? So we have the same sensory neuron. So the sensory stimuli is the same. The sensory neuron is the same in males and females. So how come that males have a behavior and females have a different behavior? Okay, that's what we were asking here. So we are going to talk about the first part, right, of the talk, neural circuits related to these, to decisions. Okay, so the behaviors why are the behavior difference? Because we have different circuits. So the circuit is not the same downstream of the female is not the same as downstream of the male. There are going to be differences. So we're going to go see what are those differences. So Ivan has a question, and do these mechanisms work the same in virgin and non-virgin female flies? No, it doesn't work the same. Okay? Virgin flies let themselves be courted. Non-virgin flies are not interested in being courted. First, they themselves are not interested. The male also recognizes them as non-fertile, as non-virgin, because once a virgin has been fertilized, it is going to smell like a male, because the male is going to leave CVA on it. I didn't want to go into all these details, just not to make it complicated. But a male can distinguish very well, between a male, a non-virgin female. Both of them smell like CVA. He's not going to use any time trying to court these two. One, because it's a male, and the other is because it's a non-virgin female that has been fertilized. There's no point in spending any time trying to court this female, because it's not going to be receptive, and the male is not going to gain anything, okay, from trying to copulate with that female. The non-virgin female that has been fertilized has a ton of sperm in herself, and she's not going to take anymore, okay? Okay. But is going to recognize a virgin female, is not going to smell like CVA, and then it's going to be courted. The male is going to say, okay, this is a female, there's no CVA on her, so I court her. Okay, Ivan? Okay, so let's move on to circuit dimorphisms, okay? So remember that we have fruitless, okay, differentially expressed, an isoform that is specific to the male. We have behaviors that are specific to the male, the courtship and the aggression, right? So, the sensory stimuli, the same sensory stimuli in males and females elicit different behaviors because the circuit downstream are different. So these dimorphisms can be in the presence or absence of neurons in the circuit. So remember that fruitless has isoforms that are common to male and female, okay? These isoforms that are common are shown in green, okay? So the female has a bunch of cells that express the common fruitless isoforms, the same as the male. However, the male expresses Fru-M and the female doesn't, okay? If you see, there are groups of cells that are present in the male and not in the female. Here, for example, there's a lot of red cells that are not present here. And here, there's not that many cells. There's another region here, right, where there's a lot of Fru-M cells that are absent in the female. In this region as well, okay. Here, there's a quite similar green, so it could be that some of the ones also that are present here are also present in both sexes, but in the male they express through M. Okay. Okay, so where else in the circuit can there be dimorphisms? Okay, so in the connectivity. So let's have a look at this circuit. Okay, so the fly brain, right, has a central part where there's there's several processing centers. One of them is the lateral horn. There's the protocerebral complex. There are some others that are not involved in this type of behaviors, right? But basically, the circuit is going to be the sensory stimuli, wherever it comes from. In this case, the olfactory system. Okay, sends the information to the sensory region for olfaction. And that information is sent to integrating centers in the brain. And then from there, because it turns out that a behavior is a motor pattern, that information is going to have to be sent, okay, to the ventral nerve cord, okay? The ventral nerve cord where there are the motor neurons. That are the ones that in the end carry out the motor pattern, okay? So any behavior is going to come from their sensory organ, is going to be processed. Then this is going to be integrated further down into the brain, and then it's going to go to the ventral nerve cord, okay? Okay, so what we have here is, let's say the circuit for courtship, okay? And you can see how there are different types of dimorphisms. One of them, if you pay attention to these little balls here, this means that the neuron, both neurons, right? So these neurons are present both in male and female. However, their arborization is different. So the fact that the arborization is different means that either they connect with different neurons or they make more or less synapses with the downstream neurons, okay? So, but there are differences in the morphology of the cell. If there are differences in the morphology of the cell, there are differences in the number in the number of synapses or the synaptic partners, okay? They're starting to be differences here, okay? Other type of differences that are more obvious in the processing part, of the brain, in the black box, or the following. There are neurons that are going to be present both in male and female in the same numbers, but there are groups of neurons that have higher numbers in males than in females. That also is a dimorphism because the signal can be quantitatively different, right? There can be more neurons, more signal, less neurons, less signal, okay? And then importantly, right, there are neurons that are only present in the male. Yeah? Male specific neurons, fruitless. Through M is a specific of males, right? So it could be regulating these differences in numbers, right? Morphology and this kind of things. Okay. Okay. So what are the mechanisms behind these morphological dimorphisms? Okay, so here what I'm showing you is like an example of a neuron that is dimorphic. So this neuron is present both in males and females, but in males it has this branch here, which means that it makes connections that, because this branch is not here in female, the female neuron does not make these connections, right? Okay, so how do you think that we go from fruitless to these changes in morphology? Anyone? We have a gene that is called fruitless that we know is responsible for this. Okay, Ada says that fruitless acts as a transcription factor, right? That would regulate the cytoskeleton. Okay, the cytoskeleton is, of course, is important for the morphology of the neurons. Okay, for the shape of the neurons. However, the other neurons respond, right? They are in an environment that they need to navigate. So how is it that they sense what's happening from outside? Okay, Dinah, yes, but what? Of course they are. Okay. Yes, so making neurons connect to other neurons. Okay, so what type of molecules are important so that neurons recognize each other? How does a neuron recognize what is happening outside? A neuron or any cell? How does a cell know what is happening outside the cell? Okay, cell surface molecules. Okay, molecules that are at the cell surface can detect what is happening outside and send a signal inside the cell so that the cell responds. Okay, so cell surface molecules. Right, so fruitless is the transcription factor that regulates the expression, right, of these cell surface molecules, right, that control the morphology and hence the connectivity. Do you guys know Robo, the slit Robo system? Okay, so what does it do, Maria? Do you remember? Okay. Yes, it's a guidance. It's a guidance system, a slit Robo. Okay, and it's a repellent, right? So Robo is the receptor, and the ligand is slit. A slit is a chemorepellent, ligand that is secreted in the midline, right? In the midline, and Robo is the receptor. And when the slit binds to Robo, the cytoskeleton of the cell retracts. So, and this way the neuron doesn't grow on that direction. Okay. So anyway, the idea here is that fruitless represses Robo1. Okay. So the neuron, this neuron is insensitive to this chemorepellent slit here and generates this kind of projection. In females, these cells express Robo, okay? So because they express Robo, the neuron senses a slit and this branch does not develop, okay? And this differential Robo expression is different in males and females because males repress, Fru-M represses Robo in the males, okay? So what happens is that, so if this is true, then a Robo1 mutant, a female that is Robo1 mutant is the same situation as a wild type male, no Robo, okay? Thus, in the female that doesn't have ROPO1, as in the male, you know, this projection appears in this site of the, you know, of the brain. Okay? Okay, so just to kind of close a little bit the idea of how fruitless, right, influences the formation and of the circuit. Okay, and why? Because of fruitless, the circuit is different in males and females. Okay, because in the end, you know, we have a gene, but we need to understand what is happening and how things are happening. Okay, so whenever you have a gene, the first thing that you want to know is whether, what is that gene? Is it a transcription factor? Is it a repressor? Is it a cytoplasmic molecule? Is it you know, a ligand? You know, depending on what the gene is, you can start thinking how it could be acting in the process that you are studying. Okay. Okay, so let's move on, right, from here to this thing. So we have solved, you know, a little bit the conundrum of why is it that the same pheromone that binds to the same sensory neurons in males and females elicits different behaviors in these two organisms. Okay, and that's because the neural circuit is different, and what makes it different is FRU-M in males, makes it different to females, because females do not have FRU-M. Okay, so now let's focus on the fact that we said that the male, you know, responds to CVA and can have two behaviors, either courtship or aggression. Okay, suppression of courtship or aggression. So how is the circuit? How is the circuit? You know, do you guys think there would be one circuit, two circuits? Anyone? This is just a voting exercise. One circuit, two circuits. Okay, so I see here a lot of people, you know, root for one circuit, one that splits in the end. Yeah, it could be two, no? In the end. Anyway, what is the answer here? Now, what we are going to address in this last part of the talk is the second part of the talk, decisions. Okay. Okay, so what is happening here is that we have the same sensory input that drives two behaviors. We have one gene, fruitless, FRU-M. Okay, so how is this managed to get two behaviors out of this? Okay, so what's happening is that there's a common node, okay, of courtship and aggression. Okay, because they are mutually exclusive, no? There's this common node that has differential integration. And depending on this integration, right, we're going to have one response or the other. Okay, so let's go here. As everything in science, things are learned, you know, step by step. Remember that I told you that we do not necessarily have all these maps, right, that the connectomics can one day provide. But that doesn't stop people to address behavior and try to identify circuits, no? So the circuit for CVA, the CVA responsive male circuit, was identified with the techniques that I presented at the beginning, okay? Tracing neurons, making sure that neurons, you know, that were in the vicinity of the dendrites of the first neuron to see if those were responding to the stimulus, and, you know, and build step by step the circuit. And here you can see how this circuit for CVA response, okay, has the other end receptor here in the olfactory system, then it goes to the olfactory processing center, and then there's an interneuron that brings this information deeper into the brain, in the processing centers, higher processing centers, and from there the information goes down to the ventral nerve core to elicit the motor pattern. Okay, so the clue here is this interrogation mark. Okay, so this interrogation mark turned out to be these cells here called P1. They are the stars. Okay, these cells are very, very important, both for for courtship and for aggression. Okay, so that's why there is a note. There is a note for these two behaviors in the circuit. Okay, so let's look at these neurons, and actually through artificial experiments and trying to identify neurons that are involved in this process, it was discovered that manipulating these neurons in the male, depending on the type of manipulation, you know, this the same group of neurons can mediate either aggression or courtship. Okay, so how does this happen? It depends on how the activation of the circuit takes place. Okay, so when the sensory input converges, so this was artificial, right? So you can choose however you activate the neuron. So when you activate the neuron, this was done with optogenetics, with low frequency, that is like light pulses with a low frequency. What that type of firing pattern elicited, the behavior that was elicited downstream of these cells, right, was aggression. See, lunging. So wing extension would be the proxy for courtship, and lunging would be the proxy for aggression. Okay, so when you activate P1 neurons with low frequency, what happens is that the animal fights. Okay, so that triggers aggression. When the activity, the activation of these neurons is at high frequency, you know, like pulses of light, very fast, one after the other, you know, that triggers courtship. Okay. There's also some aggression happening when the light is off. So let's forget about this. Okay, and let's focus on these parts here. Okay. So let's kind of go and see how does this happen, because P1s are multisensory integrator switch. Okay, so let's go here and let's see what will be the aggression outcome. Okay, so when this male comes across another male, right, is going to be recognized through both the olfactory pathway and the gustatory pathway, right, through CVA in this case and these other pheromones, you know, as this is a male. Okay, so the final activation pattern of P1 after integrating signals from here and from here is going to be low frequency, and that's going to make this male fight this other male that it encounters. Okay, that is what one would predict. What happens with courtship? Okay, so courtship, we're going to have the male that walks around, comes across an individual, right? And it turns out that the signals that it gets from the individual are from this side, okay? They are aphrodisiac signals. So the female sends all these signals through the gustatory pathway and the olfactory pathway into P1, okay? And that makes P1 fire at a very high rate. So the male decides to court the female, okay? So this is kind of the prediction, okay, of what we expect when we activate, okay? Because when we activate artificially P1 at very high, we get courtship. When we activate artificially P1 at low frequency, we get aggression, okay? However, you know, scientists, you know, look at the flies and watch movies, and they realize that not always, you know, you put a male in front of another male and there's not always aggression. There could be aggression, there could not be aggression, okay? The same, you can put a male and a female, a male and a virgin female, and not necessary that there is courtship. There can be courtship, there cannot be courtship. Okay, so how do you guys think this is explained? Okay, Victor says that there are more elements. Another is environmental outputs. Okay, so that would be environmental inputs. Okay, Alicia, Camille, because behavior is not the result of one circuit. Okay, yes, there could be integration of other things. That's also correct. Different stimuli, yes, different stimuli, could contribute to these other, other inputs, environmental inputs arriving into the thing. There is a threshold for the circuit activation. Okay, that's another possibility. So let me comment on this. Okay. Okay, so remember that I said that courtship and aggression are hardwired behaviors that are innate. Okay. Kuhn also says present on different individuals in the population. Okay, that would be also, additional stimuli. Okay. So what I was saying is that you guys need to remember that courtship and aggression, right, or innate behaviors. They are mutually exclusive. They are very important for survival. It is very important that these circuits work very well because the survival of the species depends on them. At the same time, because once you trigger them, you go all out, and we know that because once we get the P1 neuron activated, there's no going back. Okay, these behavior, the trigger of these behaviors needs to be very well controlled. Okay, because the flies live in an environment where other things happen. So let's think about a fly, a male that is very hungry because he hasn't been eating for a long time, is very, very hungry and comes across a female. It needs to make a choice, right? It needs to make a choice because I'm very hungry. Courting represents a lot of energy and maybe I need to prioritize. So I come across that, but now there is something that is more important, for me to do, which is eat, because I don't have the energy to court. I mean, it doesn't matter. I mean, I'm not ready to do that because there's another more pressing matter, okay? The same can happen whether, there's a male, comes across a female, and there's a predator, right? So he's getting, the male is getting inputs from presence of a predator, so that is going to be escape response, or courting a female, right? So, so which input is going to win? There's going to be a decision, is going to have to be made, okay? When I say a decision, the organism doesn't consciously do the decision. The neural circuits kind of, work one way or the other, to generate a behavior, a behavioral choice that allows for survival of the species, okay? So here is where we go into, again, the second part of that we have been dealing with decisions, right? So how these hardwired innate behaviors are integrated into the everyday life of the organism is taking into account, right, either internal state, the example of being hungry. When you're hungry, there's not anything sensory telling you you're hungry. It's something from the inside, or signals from the inside of your body that are telling you you're hungry, right? Internal state is a word that is, used you know, as a proxy for motivation, drive, you know, emotion. Some words that, you know, we are more used to use them in psychology rather than, you know, for humans than in animals. And, you know, maybe it's even more common to talk about these kind of emotions, drive, you know, motivation in vertebrates because they feel closer to us than in insects. Okay, so that's why we use internal state, so that people don't get upset because we say, you know, we use words that are more applied to vertebrates or humans. Okay. So after this digression, I kind of got lost of what I was saying. Well, basically, what I wanted to say is that the internal state, you know, of the flight, together with the environmental, you know, signals, you know, modulate, you know, whether these very strong circuits of courtship and aggression get switched on or not. Okay. So that's how we kind of control, you know, when to activate and not activate these behaviors, because once they are activated, there's no going back. So we need to make sure that they are activated at the right time and in accordance with what is happening inside the animal and outside the animal. Okay, and who is doing that is the neuromodulation. Okay, a lot of neuropeptides, right? So neurotransmitters or you know, mediate communication between neurons in very fast times. Neuromodulation is slower. Okay. So let's see, where does this neuromodulation take place in the circuit, because it can take place in different parts of the circuit. Okay, so here if we look at courtship versus no courtship, that is that male that encounters a female, and well, it depends on its internal state or what's happening around him, is going to do courtship or not. Okay, so this kind of modulation can be observed at different levels in the circuit. This is modulation at the level of the sensory neuron. This is a very beautiful example. The male, when it emerges, you know, we said that innate behavior is that can be done without any training. Okay, it can so it can court, but it's not going to be very efficient. Okay, it's not going to be very efficient. Why? Because the system is still not ready. Okay, so the fly can perform the behavior, but it's not going to be that successful, right? Because it's not going to recognize well females. Okay. So maturity in the fly male, sexual maturity, the peak of sexual maturity is at 1 week. Okay. And how is that controlled? Well, how do we link sexual maturity to behavior? So how do we modulate and make that these two aspects come together to make this behavior happen with more success at a particular time? Okay, so what happens is that the presence of this juvenile hormone makes the sensory neuron more sensitive. So the male has it easier to detect that the animal in front is a female when it is 6 days old than when it is 2 days old. Okay, because when there's less juvenile hormone, this neuron is not as sensitive to the palmitoleic acid. So, you know, it takes more palmitoleic acid for the neuron to fire, and here it takes less, it's more sensitive, right? So what made this neuron more sensitive to palmitoleic acid? The presence of the hormone. So this is a type of neuromodulation that is making sure, right, that a courtship, you know, is coordinated with the level of sexual maturity, because this is going to be courtship more effective at this day than at this day here. Okay. There's also other type of modulation at the level of the P1 neurons. Okay, so more in higher centers, integration centers, where this is octopamine, I think, or dopamine. I don't know if it's a D or an O. Well, anyway, there is this neurotransmitter, right? And the more of this, neuropeptide here, right, the higher the sensitivity of the P1 neurons is going to be, and the more robust is going to be the response, okay? In the case of here that we have in the male, right, in the male detection system, we have different modulators that reinforce the inhibition of courtship, like tachykinin or octopamine, okay, in this particular downstream of these sensory neurons, right? So deeper into the circuit, you know, there are neuromodulation that inhibits courtship, right? And there's neuromodulation that activates courtship, okay? In the case of aggression, the same thing happens. It is less well known where this neuromodulation in the circuit affects, okay? So we know that there is some octopamine, you know, enhancement of aggression. The presence of octopamine makes the flies more aggressive, right? Downstream of this sensory neuron, okay, somewhere in the circuit downstream of the ZR32A. And the tachykinin is also very important, but tachykinin is not clear where it acts. You know, if there is more tachykinin, the male is much more, is going to have a higher probability of engage into aggression, okay? Okay, so, you know, sometimes there are environmental aspects that make this neuromodulation, you know, push the animal. There's something in the environment that makes, you know, higher secretion of tachykinin, more octopamine, you know, and that makes the animal more at the edge of going towards one behavior than the other. Okay, in the case, for example, in this case of a male encounters another male, is he going to, you know, behave in an aggressive mode or not? Okay? It's going to depend a little bit on how the environment, the signals in the environment, or the internal state, okay, that how they modulate the circuit. Like, for example, in the case of aggression, repetitive aggression, you know, makes it more probable that in the next encounter with a male, you know, there is aggression again, okay? Because the animal is kind of, you know, now very sensitive, so the sensitivity to start aggression diminishes. As more aggression there is, the animal is primed for more aggression. The system has, it's easier to switch on the aggressive switch, right? And with less kind of stimuli, aggression gets switched on, okay? Okay, so I just wanted to kind of, you know, show you a little bit this part here. And now we have one more slide that is a little bit the summary of the concepts that we have been kind of talking about today. Okay, so we have the innate behaviors that we said are hardwired and very robust, but they need to be flexible enough to adapt to the environment and the internal state of the animal. Okay, because once they are on, there's no going back. So we need to make sure that they are on when they need to be on, okay? And not anytime jumping, you know, and getting those behaviors on. Okay, so in a sexually dimorphic behavior, the same pheromone activating the same sensory neurons in males and females elicit different behaviors in each sex. And we've seen that this is because there are dimorphisms in the circuit, okay? And that these dimorphisms in the circuit, you know, link, link the fact that there is, you know, genetic dimorphism, if you want to call it that way. You know, there is a gene or an isoform that is expressed in one sex and not the other. Okay, that matches very well with one sex having a behavior and the other sex not having the behavior. Okay. And then when there are two mutually exclusive behaviors in one sex, you know, like the example of the male, okay, that has two behaviors that are mutually exclusive, you know, either courtship or behavior. they are triggered by the same pheromone. So how is it that it goes towards one way or the other? That is going to depend on the integration. Okay, so these behaviors share an integration node that generates different output behaviors, and the outcome of these behaviors, so what behavior happens in the end, depends on the integration in the node. Remember, you know, how the cells fire, the P1 cells, and on the neuromodulation. Okay, so the presence of other neuropeptides that, you know, modulate the sensitivity of the circuit, you know, either at the sensory level or in higher integration centers. Okay, so these are the concepts that we have been talking about during this seminar. Okay, so now I'm going to show you the question that I want you to answer in the abstract format. I just want to make a comment on the abstract. Please read well the instructions. The third part where you give the answer, says 8 lines minimum. Okay, so you can write more. The first 2 sections have a maximum of lines. The last section has a minimum of lines. Okay, the total number of lines of your abstract cannot go past 30. Okay, so in the third, you're going to have 8 plus X lines up to 30 minus the lines that you have used for the first 2 sections. Okay, I'm just saying because you have plenty of space to elaborate your answer. Okay, so what about the question? So you have learned from neural circuits and decisions in the innate and mutually exclusive behaviors of courtship and aggression in Drosophila males. Okay, so now what I want you to do is to draw in analogies. God, I see that there are some kind of typos here. The question is going to be written in the Tasca in the Campus Virtual. Okay, I'll, you know, correct all these mistakes here. So what I want you to do is draw in analogies from this system. Okay, from what we have studied, hypothesize. How do you think that the mutually exclusive behaviors of feeding and foraging could be working in ants? Okay. So basically, you know, using the concepts that we have learned, that you hypothesize, right, how these behaviors of feeding and foraging could be working in ants. You know that ants, you know, eat, but then they also go and forage food, which means they go collect food to store, right? So the behavior here is I eat, you know, I go get food to eat, or I go get food to store. Okay? Camille, you have a deadline in the Tasca. It says the deadline for you to hand in this question. Okay? And you guys know, I mean, Florencia explained all this to you. You guys work in your groups, right? You work in your groups and you do this, you know, in a group, you discuss, I mean, you know, and, you know, this is very simple. We just work some concepts and now you need to translate those concepts in trying to explain another behavior in another organism, okay? So, you know, I think it's doable, and the most important, I think it's quite fun, no? Just to kind of try to hypothesize with what we learn a little bit. How do you think this could be working in ants? Okay. You guys have any questions about the seminar, about the question you need to answer? No, just hypothesize and imagine. You don't need to look for more information. You know, you could answer with what I told you, you could make an hypothesis. You know, I mean, I'm not looking for the right hypothesis. I'm just looking for something that, I mean, I'm not looking for the right answer. I'm looking for your ability, you know, with the information I've given you to make an hypothesis, you know, that in which those concepts are used correctly. Okay? No, it would not be able to. You know, it might try, you know, maybe a little part could do a little part, but not complete it. Okay? That's a good question, Alicia. That clear? Okay, good. Any other things? Okay, so now I'll finish the, you know, I just fix the things on the PowerPoint, hang it up on the Campus Virtual so you have it there. And, you know, remember to send your abstract to get, you know, by the deadline, okay? Yeah, so if there's nothing else, I think that we are done, and I guess I'm gonna exit, okay? So have a good weekend.